So now we'll move on to the Deuterostomes, and this is a group that includes us. Deutero means second, stome means mouth. So the mouth develops as a second opening. The first opening, the blastopore, develops into the anus. And we'll start by looking at the echinodermata, the echinoderms. And you might think, well, they don't really look anything like us, but they do share all of those traits that are found within the deuterostome that unite the deuterostomia. So they are deuterostomes. They are almost exclusively marine. They're sessile, which means they don't move, or if they do move, they move quite slowly. They have a center region with spokes surrounding that, and it's usually some multiple of 5. They have these calcareous plates, so made out of calcium salts, that provide an outer coating. So we could say that they have an exoskeleton, sort of, although there is some tissue that exists outside of that exoskeleton. They have what's called a water vascular system. So in this case, we have this hydraulic system. We have water that is pumped in to the system of tubes that's used to move the feet so that they can move around. They're dioecious, and what that means is there's separate girls and boys, so we have separate sexes. Gametes are released into the seawater, and there is a larval stage that results. Echinodermata is a phylum, so a large group. We're going to take a look at some of the classes now. Not all of them, just the ones that I've highlighted in yellow. So we have the Asteroideae. Aster means star. So these are what are commonly known as starfish. Of course, they're not fish. But they have a central disc, and then they have 5 arms or some multiple of 5 coming off of that. We have a second class here that includes the brittle stars, which look a lot like starfish, but they have much thinner, kind of more delicate arms. And then we have the Echinodea, which are the sea urchins and sand dollars. They don't have arms. The arms have kind of fused together, but they do still have the tube feet that we see in the starfish. We have the Crinoidea, the crinoids. This was once a very, very successful group. At the end of the Permian extinction, they were greatly reduced in number, but they are still around. This is an organism that basically looks like an upside-down starfish. So imagine taking a starfish, adding a stalk, turning it upside down, sticking it to the bottom of the ocean, and having those arms wave around in the water. That's a crinoid. They're quite common as fossils. You can find them as fossils around here. And then we have the sea cucumbers. Now the sea cucumbers are kind of interesting in that they have bilateral symmetry, and so far everything we've looked at is radially symmetric. It's thought that the ancestor of the echinoderms was bilaterally symmetrical, and we still see that in the larval forms. They're still bilaterally symmetrical, but most of the groups gave that up and returned back to radial symmetry. In the case of the sea cucumbers, they probably started out bilaterally symmetrical, became radial, and then became bilateral again. The water vascular system, let's take a closer look at that. So here is that system within a starfish. On the upper surface, kind of the back of the starfish, so opposite those little tube feet, we have this madreporite, which is a little valve that opens to seawater and allows water to enter into this system of tubes so that it can be pressurized and used to extend the tube feet. So here's that isolated, again, that madreporite on the back of the animal near the anus is an opening to allow seawater into this system. The tube feet all have these little bulbs at the base. These are ampullae, that's the plural, and these are surrounded by muscle that can contract, kind of like a dropper bottle, and force water into the foot and expand it. And the little feet, of course, have little suction cups on them so that they can grab onto things. And if you take a starfish and turn it over, we're looking at the opposite side, the underside here. So the opposite side from where the madreporite is, there's these little tiny tube feet that can be extended and moved around just by pumping water around and squeezing the water using muscle. So a few images of starfish here. The typical starfish has 5 arms, 5 spokes, but we can have more than that as well. The spokes can be kind of stumpy or they can be quite elongated. Here's a poor starfish that has lost some of its arms, and they can regrow them. They're quite good at regeneration. So as long as that central disc survives and the damage hasn't been too extensive, they can regrow their arms. This one's got a bit of work ahead of it. Here's one that doesn't have 5 arms, but it does have a multiple of 5. Then we've got the echinoidea, and this would be the sea urchins. They're covered in spines, so they have these large spines that are used for protection, and they can be moved around. On the bottom, they do have tube feet. The ones with really large spines, quite often those spines have been used for jewelry, for necklaces and bracelets and things like that. If you remove all the spines, you're left with something called a test, a shell. And we can see that 5-part symmetry in the patterns that make up this shell. So here, that opening that you're seeing at the top, that would be a fleshy bit. So it's a hole, a soft area in the exoskeleton of these guys. And that's where the anus and the madreporite would be. On the other side, there's a much bigger opening, and that's where the tube feet and also the mouth would be. And sand dollars are also in this group. And once again, if we remove all the living tissue, we can see this 5-part radial symmetry. And these are living sand dollars here. So on the bottom, they would be covered with tube feet to allow them to move around. They'll move across the sand. They'll burrow into the sand using those tube feet. Now let's take a look at the sea cucumbers, which of course don't look like that. They look like this. They're quite fascinating. They are bilaterally symmetrical. You can eat them if you have to, but I've never had one. From what I understand, they're not. They're kind of chewy, from what I understand. But they have given up on that radial symmetry, and they've returned to a bilateral lifestyle. They can get quite large. Some of them have this really neat defense strategy where they'll shoot their intestines out of their anus to scare away predators. As you can imagine, that's probably pretty frightening, and then they'll pull them back in. Okay, moving on. Now we're looking at the other phylum that makes up the Deuterostomes, and that would be the chordata, and we are included in this group. What unites all the chordates together is that they have a notochord, which is a flexible, somewhat rigid rod that runs along the back of the animal. They have a dorsal hollow nerve cord. They have pharyngeal slits, so they have a structure called a pharynx. Pharyngeal means having to do with the pharynx, so they have slits in this pharynx. And they have a post-anal tail, so the tail extends beyond the anus. We talked about that before when we were talking about insects and crustaceans. In those guys, if they have a tail, the anus is right at the end of the tail. And of course, they're all Deuterostomes. Okay, so let's take a look at this group, and we'll look at the cladogram and look at some of the derived characters that they have evolved. So the chordate structures that are common to all of them include, like I mentioned, this notochord, which is this longitudinal flexible rod that's just below the dorsal surface of the animal. And this is going to be replaced by the vertebral column, specifically the body's or centra of the vertebra, we'll come back to that, in vertebrates like ourselves. We have a dorsal hollow nerve cord, so that would be the spinal cord. It is hollow in us. Our brain has some hollows inside it as well. And the reason for this is because it develops as a tube. We have pharyngeal slits. So in some of the so-called lower chordates, they're used for filter feeding. And we have this post-anal tail with its own musculature. Now, all of these elements aren't necessarily going to be found in the adult, but they might be found in the larval stage, if there is a larval stage, or in the embryo. So a lot of these things we don't have as adults, but we do have them in the larval stage. And by larval stage for us, I mean embryo. So here is your classic chordate showing those features. We've got this dorsal hollow nerve cord. Directly under that, we've got this notochord, this somewhat stiff, flexible rod. We've got a pharynx. That pharynx has slits in it. And then we've got this post-anal tail. You can see it sticks out beyond the anus. So post meaning behind or after. And this is a much nicer view of this. So this demonstrates all of the 4 primary characteristics that allow us to group chordates together. If I have a question on the exam about, what constitutes a chordate, this is what I'm asking about here, these 4 features that all chordates share at least some part of their development. Now, I mentioned we can't see all of these features in the adult in many cases, ourselves included. Here, what you're seeing is a very early human embryo. We've got that dorsal hollow nerve cord, so it develops as a tube. The front of the tube swells and becomes the brain. Within the brain, we have ventricles, which are hollow areas, cavities, and they're continuous with this central canal, this cavity that runs the entire length of the spinal cord. We have the pharynx. It has slits, so these pharyngeal slits, which are sometimes referred to as gill slits, although, they're not always used to support gills in most chordates. They're not. The ones that you're most familiar with, mammals, for instance, they're not used that way. But in things like sharks, those slits will persist, and there will be gills between those slits. Again, we have the notochord shown in red here, that flexible rod, and then we have the post-anal tail. So let's take a look at some chordates, and we're going to start with some of the simpler ones. So we have sea squirts here, and they look terrible in the lab. They look like blobs, but this is what they look like in life. They are filter feeders. You can see that there's 2 openings on each of these animals, so we have a cluster of these animals here. They have one opening that is an in-current siphon. They take water through that, and then they have an ex-current siphon, and after the water has been filtered and oxygen has been extracted, then that water will leave through the ex-current siphon. They can be quite pretty too. This is their general body plan. So you can see water coming in at the top, and then it's passing through this pharynx. So basically the adult animal is a pouch within a pouch. The inner pouch is the pharynx. It has slits, so water is going to go into the pharynx. It's going to pass out through those slits, and then the water can be dumped back into the ocean through the ex-current siphon. There is a short digestive system, and what happens is anything that's in the water, so bacteria, plankton, algae, that kind of thing, is going to be filtered out. It's going to stick to the pharynx. The pharynx is covered in mucus. It is very sticky. It's covered in cilia. The cilia create the current of water through the organism, but also trap any microbes. And the cilia will move that material to the esophagus, which would be at the bottom of the animal here. That leads into a stomach. So that's this structure here, and then that will lead into a short intestine and the anus will dump waste into the ex-current siphon. So somewhat similar to what happens in clams and other bivalves. They are filter feeders, but unlike the clam and the oyster, they have this pharynx. And they're called sea squirts because if you see them at low tide, they quite often are exposed at low tide, and you poke one, it's going to squirt you in the eye. It's filled with water, and it's a rather muscular structure, and it will contract and squirt that water at you to hopefully scare you away. Now, if you're paying attention, you're probably asking, well, why don't we see all the chordate features in the adult here? We don't. The only thing we see in the adult that is a chordate feature is the pharynx, but they have a larval stage. So what happens is we have separate sexes here. The females will retain eggs, and then they will be fertilized. Basically, males will just dump sperm into the water. The female filters out the sperm, and the sperm will fertilize the eggs, and then the eggs are released. Then they develop into what are called ascidian tadpoles. Ascidian is another name for this group. The tadpole has all the features of the chordate. It swims around. It has this large post-anal tail that's well muscularized, so it can wave back and forth. It also has these suction cups, so you can see that we have kind of suction cups, 3 of them at the front here. It swims around for a while, and then it finds a nice place to settle down, and it very, very quickly metamorphoses into this adult form. But as the tadpole, it has the tail, the postanal tail. It has a short little nervous system. It does actually have a dorsal nerve cord that's hollow. It has the pharynx with the slits. Again, it has everything that we would expect to see in a chordate. But it only maintains that structure for about 24 hours or so. Enough time for it to swim away from the parent, find a new place to settle down, and then it quickly morphs into this simple sac. The next group we'll look at are the cephalochordata. And I mentioned before that cephalo means head. So cephalization means the evolution of a head. So the cephalochordata, they do have a well-defined head region. They have a mouth. They, of course, have a one-way digestive system. They're pretty small, as you can see, but in some parts of the world they are quite numerous. You know, you can, in some parts of the world, scoop up enough of them to have a plate of them. And they tend to spend most of their time half buried into the sand at the bottom of the ocean. They are filter feeders. So you can see on the right there at the bottom, we have their body plan. This is what they look like. And they look pretty much exactly like that ideal chordate I showed you before. So they do have a post-anal tail that has its own musculature. They do have a pharynx with pharyngeal slits. They do have a notochord, and that's one thing I forgot to mention with the last one, the ascidians, the sea squirts, the larva does have a notochord within the tail, and they have a dorsal hollow nerve cord. They can swim short distances. They have blocks of muscle that will allow the tail and the body to wave side to side like an eel, and they can swim, although most of the time they're going to be embedded in the sand and filter feeding. They're also known as lanceolets. They're the ideal chordate. Basically, they look exactly like what we think the ancestral chordate looked like. They live in sand, as mentioned, at very high densities. So in some parts of the world, you can just take a sieve and just scoop them up, and there's your dinner. They have this mouth that sticks out. There's tentacles there that will help keep sand and large coarse particles out. But they draw water in to the pharynx. They filter it. The water leaks out into the surrounding space, which is known as the atrium. The water then leaves through the atriopore, which is the excurrent siphon, and anything that's been trapped on mucus within the pharynx is going to be wafted by cilia to the esophagus and onto this short little digestive tract. They are segmented, and we see that in chordates. We are segmented as well, especially as embryos. The segmentation is pretty obvious, but even as adults, I mean, we have vertebrae that are repeated over and over again. We have ribs that are repeated over and over again within the thorax. We do show internal segmentation. All right, the next group we're going to look at are the craniates. So we're getting closer and closer to ourselves and other mammals. But the craniates, these are organisms that have a well-defined head, first of all. And they also have what are called neural crest cells. That's one of their key defining features. We don't see that in any of the chordates that we've talked about so far. What you're seeing here in this diagram is how that dorsal hollow nerve cord develops. So this is the development of our spinal cord. Notice at the bottom we have the notochord. That's the stiff, flexible rod that we see in all chordates at some point in their life. And then along the back of the animal, we have this groove that's forming. So when you were a very early embryo, after gastrulation, we have this groove that develops along the back. Basically, the skin on the back folds in on itself and forms this hollow tube. And that hollow tube will become the spinal cord. It will swell at the front end, the anterior end, and give rise to the brain as well. But when it was just a groove, you had these two crests on either side of that groove, and the cells that form there, shown in green, are neural crest cells. And they're really weird. They leave that area and they wander through the body. They give rise to the bones that make up the face. They also wander into the skin and give rise to melanocytes, which are the cells that produce pigments in the skin and hair. And all of those melanocytes, incredibly, are derived from the neural crest along the back. Also, the peripheral nervous system. So we have the central nervous system, which is the brain and spinal cord, but all the nerves that come off of that also came from neural crest cells. So these are weird cells that wander around in the embryo as different tissues and so on start to develop. The other thing that defines the craniates is that they have a cranium. That's where the name comes from. This is also referred to as the brain case. It consists of the part of the skeleton that is going to directly surround the brain. So the skull cap, basically. It's not necessarily bone. It is in some craniates, like ourselves, although it could be cartilage as well. We have appendages, usually 4, not always the case, but usually. We have very advanced cephalization, so the head is very well developed. There's a number of sensory structures involved in sight and hearing and taste and smell that are focused within the head. We have a closed circulatory system, so the blood is never allowed to just seep between cells. It's always being conducted within closed tubes. And we have a well-developed heart, we have lungs or gills, we have capillaries, like I said, so the blood never leaves tubes. The tubes get smaller and smaller and smaller, but the blood is always enclosed in a tube. The dorsal hollow nerve cord is protected by a skeleton in the vertebrates, but we haven't quite got to the vertebrates yet. So we're looking at kind of smaller and smaller groups in a hierarchy. Okay, so let's go and look at this cladogram here. So we have our ancestral chordate that gave rise to our ancestral vertebrate, and then we have the development of the vertebral column, of course, development of jaws, lungs, etc., etc. We'll get to all of those features. But the first member of the craniata that we're going to look at are the agnathons. So gnathos, it's got a silent G at the beginning, means jaw, and A means without. So these are the jawless fish. Not a good clade, because of course if we went back to our common ancestor, we'd have to include everything to the right as well. So this is not really a scientific group, but we have these jawless fishes that are, well, they're craniates, but they do not have a well-developed skeleton. So living agnathons, we have some around today. Again, jawless fishes, they have an eel-like shape to them. They're the most primitive of the craniates, so the most primitive chordates that have a very, very simple head skeleton, but no jaw. So they have a cranium, they have something, cartilage, that surrounds the brain and protects the brain, and they do have these neural crest cells. They don't have any ossified bones, so os, os in Latin means bone. So they have some very soft tissues, but they don't have the deposition of lots of minerals to make it a bony substance. They have soft bones. They have, well, not bones, they have cartilage. They don't have paired appendages. So in us, of course, we have paired front limbs and paired hind limbs. If we look at bony fishes, they have paired pectoral fins and paired dorsal fins or pelvic fins, but we don't see that here. And we have two groups, and I don't expect you to know the group names. If you just know their common names, that's great for this. So we have the hagfish, and there's 30 marine species. They're entirely marine. They have a cartilaginous skeleton. They don't have vertebrae at all, and that's what they look like. They're really slimy. If you're interested, take a look at some videos of these things on YouTube. Basically, what they do to protect themselves is they make huge, copious amounts of this very, very slippery slime, so you can't hang on to them. They also like to tie themselves in knots and untie themselves to kind of slip through your fingers. They typically live in dead carcasses, so if a whale dies and sinks to the bottom of the ocean, it'll fill up with hagfish. And then we have the lampreys. The lampreys are interesting in that they have a larval form, and that larval form is generally fresh water, so they will spawn in streams and rivers, kind of the same way that salmon do, but the adult is parasitic. So the adult is an ectoparasite. It will latch on to fish, and it's got these really nasty looking teeth that it will use to chew a little hole in the fish and suck all the juices that are produced. But with the lampreys, they do actually have very, very simple vertebrae. They have these cartilaginous structures that wrap around the dorsal hollow nerve cord. So that's the mouth of these things. Like I said, they're kind of scary. They were a huge problem in the Great Lakes. So when the St. Lawrence Seaway was manufactured, when there were locks put in to allow ships to come up to the Great Lakes, that also allowed lampreys to get in, and they caused big problems with freshwater fish within the Great Lake. So here's a comparison of a few different simple head skeletons. So we have at the top there, we have the hagfish. In yellow, what you're seeing there is the dorsal hollow nerve cord, so it's expanded within the head to form the brain. And then in green, you've got the notochord. Notice that the hagfish has nothing surrounding that yellow bit. There's nothing to protect that dorsal hollow nerve cord. So it does have a head skeleton. It has the beginnings, at least, of a cranium, so it is a craniate, but it doesn't have any vertebra, so it's not a vertebrate. The lamprey does have some more cartilage there. There's cartilage that would support the gills and cartilage that supports the gill slits. So the gill slits would be the openings that you can see here. I don't know if that shows up very well. Let me try it with a red here. You have these openings here. That's where the gill slits would be, the pharyngeal slits, and then the cartilages in between help support the gills. Down the bottom we have a shark. A shark does have a very well-defined cranium, so that top bit there is protecting the brain, and there are well-developed vertebrae as well. They're just made of cartilage and not of bone. We also have these structures here that I'll come back to that support the gills. Okay, so the craniata, again, they all have some sort of cranium that protects the brain to some extent. The hagfish are craniates, but they are not vertebrates. Everything we're going to look at from now on is a vertebrate, and that would include the lampreys that we just talked about. So lampreys, again, the adult is parasitic. It's got this nasty looking mouth filled with teeth-like structures, but it doesn't have a jaw. It doesn't have any sort of cartilage or bone to support a jaw. Yeah, I don't know why you'd want to do that, but there you go. All right, moving on. Now we'll look at the gnathostomes. Again, gnathos means jaw. So they've got a jaw that supports the mouth. So gnathostomes would include the chondrichthyes, which are the sharks and rays, the bony fish, the amphibians, the mammals, the reptiles, the birds. This is how it's thought that the first true jaws evolved. So at the left there, we've got an animal that has a cranium, so it's a craniate, but it does not have jaws. Instead, we've got these structures, these pharyngeal structures, so found within the pharynx, found within the throat, that are supported by cartilage. They're referred to as pharyngeal arches, and they would support the gills. We're seeing two here, one arch, one set of cartilages in green, one set of cartilage behind that in red, and it's thought that together these two arches, these two structures, gave rise to the jaws. And what you're seeing on the right is what we have in sharks. Sharks have this jaw, this lower jaw that's made up of several pieces, and it hangs underneath the cranium. So the cranium developed first, and then the cartilages that would support the gills gave rise to the jaws. So jaws first appeared about 400 million years ago, and it's a modification again of the anterior pharyngeal cartilages, so the cartilages that are closer to the front or anterior of the organism. It's not thought to be the first two, actually. There's evidence from molecular studies and embryology that it was arches that were a little bit further back that gave rise to the jaw. Also, in all jawed vertebrates, except for some that have lost this, we have two pairs of fins, or two pairs of appendages. So looking at this evolutionary development here, we have the jawless fishes, so that would include the lamprey, for instance. They are craniates. They do have a cranium, that's shown in yellow here, and then they do have these cartilaginous arches that support the gills. The next thing that appeared was something known as a placoderm, and you can see that in the lower left. These are all extinct, but they were a fairly successful group of fishes. They have bony armor on the outside of their head, but they have a cranium, and they have, or had, a simple jaw that was just made from one of those pharyngeal arches. And then next we have the shark, which has a lower jaw that hangs off of the cranium that's composed of these cartilaginous elements that came from two pharyngeal cartilages. And we still see that exact same structure today. Now, what's really interesting in human development of all vertebrates, we see these pharyngeal arches, these cartilaginous arches appearing. So as a very early embryo, we have these little pouches in the pharynx, in the throat. We have slits that are the pharyngeal slits. They disappear, they seal over, but we have these cartilages that are remnants of these pharyngeal cartilages that would have supported gills. That all kind of goes away, but not entirely. There's some parts of the cartilage that give rise to the inner ear bones. There's some that form as a framework for the development of the jaw, so it's replaced by bone. There are bits of this that give rise to little pieces like the styloid process that hang off the bottom of the skull, and then the cartilages that make up the larynx and also make up the rings of the trachea, they're derived from these pharyngeal arches as well. So here's a placoderm, and as mentioned, placoderms no longer exist. They are extinct, but they were a very common group. Some of them got really large, as you can see in that bottom image with a nerd for scale there. That was something known as Dunkleosteus. The head was heavily armored. This was like a very, very big, probably very vicious tadpole-like fish. Let's move on to the chondrichthyes. Chondro means cartilage. Ichthy means fish. So these are fish that have a skeleton, a full skeleton with vertebrae, but they're comprised of cartilage. So that would be the sharks and the skates and rays. There's at least 750 species. They also have something called a lateral line system. This is a tube, a pair of tubes that run just under the skin of the animal down the middle, and they sense pressure. So in a shark, it's really easy to tell where the lateral line is. It runs along here. I kind of went off the shark there, but it would run along the side here and out along the tail. Basically, it runs between that dark upper surface and the white lower surface. It's also interesting that they have that coloration. It makes sense. If you're underneath a shark and you're looking up, the white is going to be seen against a bright background. So we have sunlight coming down, of course. And if you're looking down, if you're above the shark, the darkness on the back is going to help camouflage it against the dark bottom, the depths of the ocean. So this is a camouflage feature. So this lateral line system, again, it's under the skin and it senses changes in water pressure. It's used for hunting, so if something is thrashing around in the water, they can sense those pressure waves that come through the water. Some sharks are viviparous, that means they give rise directly to free-swimming young. Some are oviparous, that means they lay eggs. So ovi means eggs. Some are ovoviviparous, which is a combination of the two. There's snakes that do this as well. It's where the egg breaks during its laying. So for instance, in garter snakes, they have a very, very thin-walled shell around them. It's clear, and as they're passing out of the mother, it actually breaks. They have something called a cloaca. The cloaca is the exit for the digestive system, the urinary system, and also the reproductive system. So all of those systems have one common opening. And cloaca, funnily enough, is Latin for sewer, which makes sense because we have all this stuff coming out of one hole at the end of the animal. And this is the case for most vertebrates. Mammals are the exception. We don't have a cloaca, although we do when we're embryos. Here's the largest current living fish. This is a whale shark. Very, very large, but actually a filter feeder. So they will swim around in usually fairly shallow depths, and they will filter in plankton. So moving on, we'll take a look at vertebrates that have a bony skeleton. So chondrichthyes, the sharks and the rays, are fully vertebrates. They do have properly formed, fully formed vertebrae, and they have a fully formed cranium as well, of course, and jaws and all that stuff, but it's made of cartilage. Bone, of course, is a bit more rigid because it has a lot more mineral deposits. As mentioned, os, is Latin for bone. An ossified tissue is a tissue that has considerable deposits of minerals, calcium carbonate, for instance, that makes it far more rigid than cartilage and other connective tissue. Your long bones, your humerus and radius and femur and tibia, etc., start out as cartilage. In the fetus, they are cartilage, and then that cartilage is replaced by bone. It becomes ossified. Bone is a living network. There are living cells within bone called osteocytes, and they maintain the bone. And the matrix that surrounds them, the material that surrounds them, is about half protein, mostly collagen, and half mineral salts. So we have this combination of proteins that are rather stretchy and they are capable of resisting tension. And then we have these solid salts that resist compression. And together, that makes for a very strong substance. At the top here, we have a very young fish, a fish fry, as they're usually called. We have a notochord, that's a flexible rod that's found in all chordates at some point in their life history. And what happens in vertebrates where we have a bony skeleton is that that notochord is replaced by the body, also known as the centra, of the vertebrae. So that big chunk at the bottom there, that is going to replace most of the notochord. Now, in between the vertebrae, we do have this somewhat flexible disc, the intervertebral disc. You might have heard of a slip disc before. That's a leftover of the notochord. And the first group of bony vertebrates that we'll look at are the osteichthyes. So again, os means bone, ichthyes means fish. These are the bony fish. There's lots of species. They've been quite successful. And we have three classes that we'll look at very briefly. They have an ossified skeleton. Again, that just means there's lots of minerals that have been deposited. So the skeleton is in many ways similar to the cartilaginous skeletons that we see in the sharks, but it's been ossified. It's been made harder by the deposition of minerals. We have 5 pairs of gills, but they're covered by something called an operculum. So in a bony fish, we have this big flap here that covers those gills. They also have something called a swim bladder, and this allows them to function kind of like a submarine. So they can take dissolved gases out of their blood, pump it into the swim bladder, and then raise up in the water. So they can control what level they're sitting at in the water. They can control their buoyancy. So let's take a look at a typical bony fish. This is something that you might find in the supermarket, a trout or a perch or something like that. They have this kind of anatomy to them. You can see that there's a swim bladder there. This is a thin-walled pouch that can fill with gas and allow the fish to rise up or sink within the water column. We have the lateral line, so we have a line that runs down the length of the fish, and this is a fluid-filled tube that can sense pressure. Incidentally, we still have the lateral line in us, but it's become restricted to the inner ear and it senses equilibrium. We have a long kidney, as you can see at the top there. We have a fairly simple digestive system. This is a fish that's eating a lot of protein. It's eating insect larvae, that sort of thing, which is pretty easy to break down. We have the heart, which is further forward than you might expect. It's simpler than the heart that we have, but we'll talk about that in a moment. And recognize that the basic body plan of a bony fish is still the same as the basic body plan of any chordate. We have this notochord that's going to be mostly replaced by the vertebra, and we have this hollow dorsal nerve cord, the spinal cord, that becomes a bit swollen in the front, the anterior of the animal. We have a pharynx. We have pharyngeal pouches and slits. Now this is all covered by that operculum. And we have this digestive tube that ends forward of the tail. So we have a post-anal tail. Fish gave rise to the tetrapods, and the name tells you all about them. Tetra means 4, pod means foot. So these are animals with 4 limbs. Now, the bony fish do have 4 appendages as well. They have 2 pectoral fins. Those are in the front. They have 2 pelvic fins in the back. They also have a dorsal fin, but we're kind of focused on those bottom or ventral appendages. The first tetrapods to appear were the amphibians. So these are, as the name suggests, organisms that can live in water and on land. And then we have, of course, the reptiles, the birds, the mammals, and we'll come back to all of those. There are some tetrapods that have lost their limbs, or maybe lost just one set of limbs. So for instance, we have the snakes that have lost their limbs for the most part, although in boas and pythons we still have hind limb buds in the embryo. So let's take a look at the first class of tetrapods to appear. We have the amphibia. Amphi means both ways or two ways, so these can live on land or they can live in the water. Most amphibians need to be kept moist. They need to be in a wet environment, and they also breed in a wet environment. Water is absolutely essential for their breeding. Now, the one that I have on the screen right now is a very unusual amphibian. This is something known as a caecilian, and it's sometimes referred to as a slow worm, something you might find in Mexico and South America, for instance, but they do have a fairly wide distribution. It looks like a worm, but it is a vertebrate. It has a vertebral column. It has vertebrae. It has tiny little eyes. It lives in the soil, this particular species. It's lost its limbs, but it is derived from the first tetrapods. So we have salamanders, we have frogs, we have caecilians, they're kind of the odd ones out. We have about 5,000 species or so, and again, they are the first tetrapods. They have external fertilization, usually. Now what that means is that the females will lay eggs, and then the males will release their sperm into the water, and the eggs are fertilized outside of the female. And metamorphosis occurs within the water. So we have larval stages, we have tadpole stages. The tadpole is devoted towards eating and growing, and then it metamorphoses into an adult. Now that's not always the case. There are some frogs that skip that step, which is interesting. There are a few amphibians where fertilization does occur internally. Caecilians, for instance, they have a larval stage. They have a tadpole, but it lives inside the mother. And they actually quite often eat each other inside the mother, which is kind of frightening. But yeah, your typical amphibian would be something like the frog. You can see a male and female here that are engaging in sexual reproduction. The female is a large one in front. She has laid lots of eggs. The male is grabbing onto her, and the eggs have already been deposited, but he's releasing sperm, and that's going to fertilize the eggs in the water. So for the most part, amphibians are very dependent on water for fertilization. The eggs do not have a shell of any kind. They have kind of a layer of goo or jelly around them, and they have to be kept damp. So where did the first amphibians come from? Well, as is always the case, we don't have time machines. We can't go back and say that for sure this group gave rise to this group, etc. We have to make these cladograms and kind of group things together and try to look for patterns and trends and so on. But if we look at fish, we have three big groups that we'll talk about briefly. We have the ray-finned fish, which are things like perch and salmon and so on. And you're seeing that in the first diagram of a fin there, and they're called ray-finned because they have these tiny little bones that are kind of ray-shaped. They radiate out from the girdle, the attachment to the axial skeleton. Then we have lungfish, which have an interesting pattern of bones, but the really interesting one is the lobe-finned fish, the last one there. If you just know it as lobe-finned, that is fine. Notice that they have a humerus, a radius, and an ulna. There are living members of this group, the coelacanths. We'll talk about them in just a second. But this is the group of fish that gave rise to the very first amphibians. The very first amphibians were basically fish that could come up on land for short periods. There are lots of fish like that today. There's a lot of freshwater fish, catfish for instance, that if their pond dries up, they can get up and walk across the terrain, walk across roads. This is something that happens in the south of the US, where you have these migrations of catfish and they cross highways and cause havoc by doing that. But they can cross roadways and so on and find a new pond that's not dried up. So you can see how having this ability to move from pond to pond might be advantageous. Now this is the coelacanth here. Really quite fascinating, because this is something that's been known about from fossils for a very long time. No one realized, at least biologists didn't realize, that this thing was still around. So we have fossils of coelacanths that were discovered back in the 1800s, and they were well studied, and it was realized that, okay, these are the missing link between fish and amphibians. And then in the 1930s, it was realized that native individuals in Polynesia, for instance, had been fishing for these things for a long time. They still exist. They're in the very, very deep ocean. In fact, you can't bring them up without killing them. They live under very high pressure in very, very deep waters. But the coelacanth is a living fossil. These things are still around, and their anatomy suggests that they are remnants of the missing link between fish and the first amphibians. The first amphibians are interesting in that they had more digits, more fingers and toes, than we see in vertebrates today. So if we're talking about reptiles, if we're talking about mammals, we have 5 digits in most of those vertebrates, but the very first vertebrates probably had quite a few more. For whatever reason, the forms that had fewer, that had 5 digits, were the ones that really took off and gave rise to the first really successful tetrapods. And what you're seeing in the image here is an artist rendition of one of those very, very early amphibians, and you can see how fish-like it is. Again, we can't say for sure that one fossil species gave rise to another, etc., but there are so many transitional fossils that show this change, this trend over time. At the bottom there on the drawings, you can see something that's very, very fish-like, and then you can see how that changed into something that is more terrestrial, more amphibian-like. We have really cool footprints, fossils of these very, very early steps onto land. So what you're seeing at the top there is a fossil trackway of an early amphibian that wandered up onto land, probably just to move from one pond to the other. Down the bottom there, we're seeing this trend that occurred in the development of the limbs of amphibians. So we can see this pattern of bones that developed in the lobe-finned fish and ultimately gave rise to what we see in the first amphibians, and then what we see in living tetrapods today. The next group we're going to look at are the amniotes. Now, the amphibians were and still are restricted to rather wet locations. They need standing water, fresh water, to reproduce, and that's because they have these eggs that are soft-shelled, if they have any shell at all. Typically they're just covered with a gel-like layer. They have to be kept moist, and fertilization is entirely external with only a few exceptions. So the sperm needs to swim through the water, get through that layer of goo, and fertilize the egg. So amphibians couldn't wander too far from fresh water, and that kind of limited their success. Amphibians today are in a bit of trouble. I think I mentioned this in class. They are very, very sensitive to pollutants because they are so dependent on fresh water, and because their eggs are very permeable, and because their skin is very permeable. So amphibians do get some of their oxygen and so on from their skin. It actually diffuses through their skin. And they're also very, very sensitive to a lot of parasites. There's been an issue lately in the last few thousand years probably with a lot of citrid fungi. If you remember, those are the simplest fungi, the most primitive fungi. For some reason, they seem to be doing very well at infecting amphibians lately. In any case, in order to move onto very, very dry land, animals needed to evolve a shell, a sturdy shell that would protect the egg from drying out. And the very first chordates that did that are known as the amniotes. So amniotes developed this hard outer layer to their egg that protected the egg from desiccation, from drying out. The amniotes include the reptiles, the birds, and the mammals. Amphibians gave rise to the first reptiles. The reptiles are responsible for the evolution of this new invention, the hard-shelled egg. So the shell is made out of calcium carbonate. It's permeable. It allows air to move back and forth, so oxygen to come in, carbon dioxide to go out, but it also resists desiccation, drying out. And this allowed vertebrates to move up onto land away from water. So now eggs didn't have to be laid in water anymore, and this allowed them to move into drier and drier locations. And of course, many reptiles are very, very good at living in very dry places. Now, the amniotes would include those original reptiles and the reptiles that are around today, but also birds and also mammals, which might seem kind of strange because mammals do not lay eggs, but we'll come back to that. They evolved from an amphibian ancestor, as mentioned, and the egg development that we see in amphibians limited the amphibians. They meant that the amphibians were dependent on water. Having this breathable, permeable, hard-shelled egg allowed for vertebrates to move away from the water. The shell is made of calcium carbonate. It helps retain fluids within the tissues of the egg and the embryo. And then also we had the evolution of what are called extra embryonic membranes. These are membranes that are produced by the embryo that allow for gas exchange, waste storage, and also allow for the storage of nutrients that are contributed by the mother. So let's take a look at the egg that we see in reptiles and birds. It's quite a bit different from what we see in amphibians. In amphibians, there's just a layer of goo, this gel-like layer, and in the middle of that we have our zygote that develops into our embryo, and that's it. There are no extra embryonic membranes. In the reptiles and the birds, we have the development of these extra embryonic membranes. These are membranes and structures that are produced through the efforts of the embryo mainly, and also through contributions of the mother. So first of all, around the outside we have the shell. It's made out of calcium carbonate. It is breathable, and if you look at a typical egg, and this would include a chicken egg, at one end, the rounder end, we have an airspace. And we have exchange of gases at this end of the egg. So if you hard boil an egg, you probably know that at the rounder end of the egg, there's a little gap there. And that's where gas exchange would occur. So oxygen is coming in, carbon dioxide is being released, and of course that would be to supply the embryo with the stuff that it needs. Now the embryo itself is surrounded by an amniotic sac, and within the amniotic sac is fluid. What I find really fascinating is that the pH of that fluid, the salinity of that fluid is very, very similar to the ocean water that these vertebrates first evolved in. That's really what homeostasis is all about. within our cells, we maintain the primordial ocean in which we evolved. And that's what's going on in an egg. That amniotic sac is a little pouch of ocean that's maintaining the development of this embryo. So even though it's developing on dry land, maybe in a desert, there's a little pocket of ocean in which that embryo is forming. The embryo is connected to a yolk sac, and this is contributed by the mother. The yolk sac contains lipids mainly, and the lipids are a storage molecule. They store lots of energy that the embryo can feed off of, and you can see that there's blood vessels that lead from the yolk sac to the embryo. We also have something called the atlantos, which is for the storage of waste. Because of course, if you're in a sealed egg, you can't get rid of the waste. You have to put it somewhere. So it goes into that pouch. So we have all of these specialized membranes that help support the embryo. A couple more things about that egg. The albumin is made up of protein. So it's a mixture of amino acids that help provide amino acids for the development of the embryo, but also provide a further form of nourishment. So we have the yolk, which is mostly fat, mostly lipid, and then we have our source of protein. Also, if we take a look at the connections to the yolk and also to the atlantos, we have these blood vessels that go back and forth between these structures, and this is an umbilicus. So this is exactly the same structure that we see in the placenta that evolved later in mammals. We see an umbilicus, we see a structure that is collecting waste, and in mammals it passes it on to the mother. We have a structure that is collecting nourishment, and in mammals that's connected to the mother as well. So basically mammals are amniotes because the egg is retained within the mother. So in a placental mammal, the structures that we see here also exist, but they form part of the placenta. We have the amniotic sac that's filled with amnion, and then we have our yolk sac and our chorion, which form the placenta. So let's take a closer look at that first group of amniotes, the amniotes that evolved directly from amphibians. These are the reptiles. They're not a true taxonomic group because we should really include the birds in this group. We'll come back to the birds. They first appeared about 300 million years ago. There's about 6,500 species. They're still doing pretty well, but of course their heyday was back in the time of the dinosaurs. We have turtles, lizards, snakes, and crocodiles, and also alligators and so on, other members of that group that are still with us today. They have scales that help protect them from desiccation. They protect them from drying out. And the scales are filled with a really tough protein known as keratin. Now that's interesting because our skin is filled with keratin. Our fingernails are made of keratin. Our hair is made of keratin. They have lungs for gas exchange, and the lungs evolved from the swim bladder that we see in fish. So there are a number of fish, probably the best example would be the lungfish, that use that swim bladder to extract oxygen from air. And if you've kept tropical fish, you might notice that occasionally they go up to the surface and they gulp in some air. They do that to kind of recharge that swim bladder. But there are some fish, a number of catfish, for instance, that can take in air from the atmosphere, take that into their swim bladder, and then extract oxygen from that. So there are many species of fish that can actually move from pond to pond, and as they're doing that, they gulp in air and they gulp that into their swim bladder and they extract oxygen from it. Also, reptiles are ectotherms. So ecto means from outside, therm refers to temperature. So they need to regulate their temperature by regulating their exposure to sunlight. If a reptile is too cold, it'll go out and sun itself, basically. And we have a fairly diverse group here. We have turtles and tortoises, we have lizards, we have snakes, and we also have the crocodilia and alligators. So continuing on here with the amniotes, again, we're looking at reptiles and birds, but we're also looking at mammals. Reptiles would just include the turtles, the snakes, tortoises, crocodiles, and again, it's not a true taxonomic group because we're not including the birds, whereas we really should if we're trying to form a proper clade. The age of the reptiles was the age of the dinosaurs. So there was a period of time where all of the large animals on the planet were dinosaurs. We had dinosaurs that were quite small too. We had dinosaurs that were the size of chickens and smaller. So most of the niches, most of the roles that we find in ecosystems were occupied by dinosaurs, were occupied by reptiles. So we had this period of time where reptiles were definitely the most obvious large organisms on the planet. That came to an end at the end of the Cretaceous. So 65 million years ago, we had an impact of a large asteroid. As we've talked about before, there's lots of evidence for this. We have this layer of iridium around the planet. Iridium is pretty rare on Earth, but it is quite common in asteroids and meteorites. So really good evidence that we had a major impact that would have thrown huge amounts of dust into the atmosphere, would have caused worldwide forest fires. We probably had a few years of very, very dark skies, and terrestrial ecosystems suffered as a result. Now, in the shadow of the dinosaurs were mammals. Mammals appeared quite a long time ago, 150 million years ago. So for many, many million years, mammals were there, but they were outcompeted by these reptiles. And then they finally got their chance. So out of the shadows, we had mammals taking their turn. So let's move on and look at the mammals. Mammals evolved from reptiles, specifically from a group known as the therapsids. These are warm-blooded reptiles that don't exist anymore. So they would have had a very high metabolic rate. Instead of just relying on their environment for warmth, they created excess warmth through their metabolism. Currently, we have about 4,500 species of mammals, and in the broader sense, that's not a whole lot, really. When we think about animals, we tend to think about the cute, fuzzy ones, but really they represent a pretty small percentage of the animals. Mammals are viviparous. That means they give birth to live young that aren't in an egg, with a few exceptions. So there are some current living oviparous mammals. Those are mammals that lay eggs. So that would be the platypus and the echidna. Mammals have mammary glands. They produce milk. That is one of their distinguishing features. That and the fact that they have hair. And the hair is made of keratin. In fact, hairs are modified scales. They also have differentiated teeth. And what I mean by that is if we look at our own teeth, we have incisors, we have canines, we have molars, we have specialized teeth that do specialized things. They cut, they grab, or they crush. Whereas if you look at a reptile, the teeth are all kind of cone-shaped. They all look very similar. Let's take a look at those teeth. So in a typical carnivore, like you see in the skull at the top there, we have canines that are used to grab onto the prey while it's struggling and fighting until the animal can subdue it. We have incisors that will cut off chunks of flesh. We have premolars that are kind of like scissor blades that will come together and cut off chunks. And then we have molars that will grind stuff up. That's for a carnivore. If we're talking about a herbivore, then we have incisors that can pluck up blades of grass or leaves, and then we have molars that will grind the stuff. And if we're looking at an omnivore, something that eats everything like ourselves, then we have kind of a mix there, but we still have specialized teeth. We still have those canines, we have those incisors to cut things into chunks, and we have the molars to grind stuff up. Compare that to a reptile. We have these cone-shaped teeth. There's typically lots of them. They all look very similar. The therapsids, the ancient mammal-like reptiles that gave rise to mammals, were a mix between reptiles and mammals. They looked like reptiles, but they were warm-blooded. They had a high metabolic rate, and they produced heat, and of course that increases the rate at which you can move and do stuff, so that is quite advantageous. And we do have lots of fossil evidence of these creatures. Okay, so looking at mammals, we have our ancestral mammal, and then we have an early branching point here. One branch gave rise to the monotremes. And these are egg-laying mammals, and we still have two examples. We have the platypus and we have the echidna. The next groups that branched off were the marsupials, and they had pouches, so they, of course, still exist. They're very prominent in Australia. We talked about that before. Australia separated off just before the placental or eutherian animals evolved. In most parts of the world, the eutherians outcompeted the marsupials. But in Australia, again, because it was isolated, the marsupials did very, very well there. Marsupials have a pouch. They give birth to, well, from a eutherian point of view, a rather underdeveloped embryo that then is going to develop further in the pouch. It's going to feed on milk in the pouch. In the placental mammals, the eutherians, development is taken further within the womb. So monotremes, they still do exist. I mean, this was a much larger group at one time, but these are mammals that lay eggs. They are the missing link between reptiles and eutherian mammals. So we have the platypus, we have the echidna. They will lay eggs, they will sit on those eggs and keep them warm, as a reptile or bird might do. The other thing that's quite interesting is they don't have nipples. That's something that we see in placental mammals. They have these glands that lead to one duct that's found in the nipple, and milk is produced. Now, milk is modified sweat, and what the echidna do, and also the platypuses, is they will produce this thick, viscous sweat all over their chest, and when the babies do hatch, they just lick that off, so they don't have these dedicated nipples. So the duck-billed platypus, really, really weird looking creature. In fact, when they were first discovered by Europeans, and Europeans brought specimens back to Europe from Australia, no one believed it. No one believed this was an actual organism. They thought that they had taken a bill off of a duck and stitched it onto a dead beaver or something to that effect. Another big change, looking at amphibians and fishes and looking at reptiles and looking at mammals is the way the heart is structured. So in the fish, we have a heart that has two chambers to it. We have an atrium that receives blood, and we have a ventricle that pushes the blood out. There's just two chambers, these two muscular chambers in a tube. And if you think back to the invertebrates we talked about, they usually just have one chamber. They have one swelling, and they have muscle around that. In mammals, jumping to the other extreme, and also in birds, we have two separate atria, two separate ventricles. We have one side of the heart that deals with deoxygenated blood. We have another side of the heart that deals with oxygenated blood. So in this diagram, deoxygenated blood is blue, oxygenated blood is red, and the two do not mix. This is very efficient. In reptiles and amphibians, we have a three-chambered heart. We have two separate atria, but the ventricles are fused together. We have one ventricle, basically. That allows for some mixing, but then again, reptiles and amphibians aren't nearly as active as mammals and birds. They don't require quite so high levels of oxygen. If there's a bit of mixing, it's not a big deal. The vertebrate brain has changed quite a bit too between these different groups. If we look at the fish, especially so-called lower fishes like sharks and lampreys and so on, most of the brain is dedicated towards smell. That's how they sense things. If we look at the bird, the cerebellum, which is the little bit at the back of the brain that's involved in balance, is very large, because birds, of course, have to worry a lot about balance and maintaining flight patterns and so on. And if we look at mammals, the cerebrum, which is the part of the brain that's involved in higher thought processes, cognition, that's the part of the brain that's more advanced. And on to humans, who, well, because we're a bit biased, we tend to consider the pinnacle of evolution. I mean, we're pretty generalized, really. We've talked about this before, but if we were to trace back our common ancestor, we go back to about 4 million years or so, we have humans, pygmy chimpanzees, and gorillas all coming from a common ancestor. We go back a bit further to about 6 million years. That includes orangutans as well. We go back to about 10 million years ago, and then we're including gibbons. Gibbons would be the outgroup here. But just keep in mind that the old myth that, you know, if humans evolved from chimps, why do chimps still exist, makes no sense, because this is a branching process, and as species diverge, they change as well. So humans have changed a lot in that last 6 million years, but so have pygmy chimpanzees, so have gorillas. There was a period of time where we had a number of different hominids that coexisted. So our genus is Homo, which means man. The current species is Homo sapiens, sapien meaning wise, so wise man. But there were other Homo species. There were also Australopithecines as well, which were a separate genus, and they did overlap. So how these different species interacted is not clear. The problem, of course, is that humans were not a terribly common and successful group in the beginning. So we had maybe 1 million hominids in Africa for quite a long period of time. We had these different species, different genera as well, that were coexisting, competing with each other, probably being rather nasty to each other as well. It's possible that Homo sapiens wiped out Neanderthals. It's also possible that Neanderthals just became absorbed into Homo sapiens because they were fairly closely related. But again, it's impossible to tell, and we probably will never know for sure. And that's because this evolution occurred in an area that was largely erosional. Sediments were being eroded, they were being washed away, they weren't being deposited. So we don't have that many fossils because of the erosional situation, and also because of the fact that, well, humans really weren't that common when all this interesting stuff was happening. Anyway, humans first appeared in Africa. We had Homo erectus, which was the first species that started to explore the world, heading up into Asia. So we had waves of migration. We had waves of migration of Homo sapiens into Asia, and then on into Australia, and across the Bering Strait into North America. So during the last ice age, a lot of water was locked up within the glaciers that were covering the northern continents, and that lowered the sea level so that we had these individuals that could just walk across the Bering Strait. So traditionally it's been thought that humans migrated into North America about 15,000 years ago. It might have been much earlier. There's some people that think maybe it was as early as 40,000 years ago. But regardless, humans migrated through North America down to South America and gave rise to the indigenous populations that we see there. The last group that we'll look at are the aves, the birds. Now, again, birds should really be considered as reptiles. They should really be considered as living dinosaurs. They're very, very closely related. The velociraptors from Jurassic Park. These were warm-blooded dinosaurs that came from the group that gave rise to birds. And birds are warm-blooded. They have very high metabolic rates, which makes sense. I mean, flying takes a lot of energy. They have a higher metabolism generally than mammals do. Now, some birds, of course, have lost the ability to fly, such as the penguins that we see here. So birds are considered today as living dinosaurs. They have a number of adaptations that appeared for flight. They have hollow bones. They have air sacs that actually go into those bones so they can extract more oxygen from the air they breathe in. They have feathers. Feathers are very highly modified scales. If you look at a bird, if you look at its feet, it does have scales on its feet, like a reptile would. But we talked about this before when we talked about Darwin. Darwin had noticed, of course, that there are several bird species, especially penguin species, that have feathers on the feet. So if you actually tweak a few genes, some of the master genes, then you can prevent scales from developing on the feet, and instead you can have them develop into feathers. So feathers are highly modified scales. Hair is also a highly modified scale. We have no teeth in birds, and that's to save weight. Teeth are heavy and they would throw the bird off balance. We have a very large sternum or breastbone, and that's for the attachment of very powerful flight muscles. They are endotherms, which means they generate their own heat. They don't have to get heat from their environment, and that's because, again, it takes a lot of energy to maintain powered flight. So did dinosaurs really go extinct? Well, not really. What you're seeing here next to the duck, so in the upper right, is Archaeopteryx. Archaeopteryx is an ancient bird-like reptile. It's probably not the direct ancestor of birds, but you can see that this is something that had many bird-like features and many reptile-like features. It had claws on the wings. It had teeth in its mouth. And you can see a fossil of this below. So there are many, many fossils that have been found of things that are definitely reptiles that do have feathers. And here's Archaeopteryx again. Again, it's not the direct ancestor of birds, but it's an interesting mix of reptilian and also bird features. So we have a skull that has teeth. That's a reptilian thing. We have a tail, shown in purple here, that's also very reptilian. We have ankles that are very reptilian. So if you remember in the lab, we saw that the ankle bones have mostly disappeared. We have digits that attach directly to the tibia. Not the case here. We have a shoulder girdle that is similar to reptiles. We don't have the furculum, the wishbone that we see in birds, and we have claws on the wings that are very similar to what we would expect in a reptile. We don't have those weird bones that developed from the fusion of different digits and arm bones that we see in birds. But we have feathers. We have very clear imprints of feathers. So this is a very famous fossil. There's several of these that came from Germany, and they came from a very ancient lake that had very fine sediments, silt, clay, that preserved structures really well and preserved feather imprints. And another view of what this critter might have looked like in life. Probably was not a very strong flyer. There have been a number of small dinosaurs that have been re-evaluated lately. Taking a close look at the fossils shows that there are some very, very fine, wispy, feather-like structures. So it is possible that some of our favorite dinosaurs might have had a downy coat on them, a coat of these downy feathers that would help trap heat, because they likely were endotherms. They likely created their own heat and retained that heat and had a high metabolic rate and high levels of activity. So our topics here, again, the chordata divided into 3 subphyla. So chordata is a phylum, and then we have our subphyla. The thing that defines the chordates is that they have a pharynx with pharyngeal slits. They have a dorsal hollow nerve cord, they have a notochord, and they have a post-anal tail. So the anus is not at the end of the tail as it would be in an invertebrate, most invertebrates. Yeah, and I mentioned before, imagine if your cat had its anus at the end of its tail. That would be kind of weird. The pharynx originally evolved for filter feeding, but of course that's not something that most of the chordates do anymore. We have jaws that popped up in the Gnathostome. The tetrapod limb evolved from a lobed fin, so we had lobed finned fishes like the coelacanths. Originally we had more than 5 digits, but the particular ancestor that won out and proved to be very successful had 5 digits. So all tetrapods that we see today have 5 digits, or they have something that very obviously evolved from that. So for instance, the horse has lost most of its digits, the snake has lost all of its digits, but they did appear from this very successful 5-digit ancestor. We have an amniotic egg that appeared in the reptiles that allowed them to be less dependent on water for reproduction. Reptiles gave rise to mammals and also to birds, and birds are considered to be living dinosaurs. Note that the echinoderms are deuterostomes, but they are not chordates. They don't have that pharynx. They don't have any of those other traits that we've discussed. And here's the terminology. And I am sorry that we had to kind of rush through this, but hopefully you found this quite interesting.